Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Happy New Year. We hope you had a restful holiday season. On our end, we're excited to be back with brand new Dr. GPCR podcast episodes. Before we dive into this episode, we are excited to launch the Dr. GPCR University program with the goal of providing courses and content on all aspects of GPCR research to support our community. To kick off this new initiative, we partnered with Dr. Terry Kinakin, who will be teaching a course entitled Applying Pharmacology to Drug Discovery starting February 8th. The course will be held every Thursday through February 29th, starting at 10 a.m. Eastern Time. Spots are limited to 20 in total, and the deadline to register ends on February 1st. If you register and join us, you'll get 4 live Zoom sessions with 1-hour lectures and 30-minute Q&A each time, a 30-minute one-on-one meeting with Dr. Kinakin according to his availability, access to the cohorts group in the Dr. GPCR ecosystem, where you can find Dr. Kinakin himself and your classmates. You'll get access to the course materials and recordings. You'll get a completion certificate signed by Dr. GPCR and Dr. Kinakin. And if you'd like to take advantage of this, we're also giving you a 1-year Dr. GPCR Ecosystem Premium membership, which is a $250 saving, which gives you access to the Dr. GPCR Ecosystem contents and perks, including our forums, private groups, and to all ecosystem members. And this way you can revisit the course contents at any time and take advantage of visiting the other course contents later on. To get more information and register for the course, join the ecosystem for today for free. What are you doing April 22nd and 23rd? Join us for the inaugural Endocrine Metabolic GPCRs meeting taking place in Birmingham in the UK. This event will bring together scientists from both academia and industry to exchange data, technologies, and ideas in the field of GPCRs in the context of endocrine and metabolic receptors. We have a special offer for the Dr. GPCR community. Visit our website to get more information. The key topics covered at this meeting will be the latest GPCR signaling methodologies, metabolite sensing GPCR biology, and novel endocrine mediators and GPCR activation structures. This meeting is organized and we're partnering with Biospecifica to bring you this meeting. This meeting is also endorsed by the Society of Endocrinology. Mark your calendars again for the next season of the Dr. GPCR Symposia, and mark your calendars for our first event of the year, which is March 15th. This first symposium will cover the topic of GPCR activation and signaling. We will have talks, and we will also be accommodating posters on Zoom in breakout rooms, so stay tuned. If you'd like to give a talk at any of our events, or if you'd like to be my guest on the Dr. GPCR podcast, please email us at hello@drgpcr.com. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. And this afternoon, I'm very excited to have with me Dr. Daniel Isom. And I almost got it right. Dan, welcome to the podcast.
Oh, thank you. It's a pleasure to be here.
I'm
looking forward to doing this.
Thank you. I was so focused on saying your last name properly that I messed up your first name. All right.
It happens.
We were just talking about this before we hit record, and I was mentioning as well that my first and last name have been butchered a couple of times, but it's okay as long as we know why we're here, what we're doing. The rest doesn't matter. All right, exactly. Dan, let's start at the beginning. Can you please introduce yourself? Tell us who you are and what do you do?
Sure. My name is Dan Isom. I'm an associate professor with tenure in the Molecular and Cellular Pharmacology Department at the University of Miami, Miller School of Medicine, the hot Miami, not the Miami in Ohio. I have appointments we have a cancer center here, an NCI designated cancer center, and also our Institute for Data Science and Computing. And I run a lab that does leading edge research in GPCRs and other proteins using computation and experimental work.
This is really cool. And I promised I would ask you, where does your last name come from? Does it have any meaning or any Where does it come from, basically? I think I'm tired. It's Thursday night and I'm
Yes,
getting
I'm with you. It's 5 o'clock. We're whoo. Um, Isom, I don't exactly know. I think the origins were Isham, which is a European name. But when I was younger, I was told it was also derived from Native American heritage. There are also in Mississippi, there are also people with Isom as a first name that I found. So it's kind of mysterious. It's kind of rare. It's kind of unusual.
yeah.
But despite against all odds, there's another Isom in science
Hmm.
at another UM, Michigan,
Okay.
and she's in the pharmacology department, and she is the chair. So when you go to Google Isom, UM, you get me at the University of Miami and Lori Isom at the University of Michigan. And we're both in the
Wow.
pharmacology department.
Interesting. And you have absolutely no relationship other
No
than being a
relationship whatsoever. I met her, I came to know of her because the NIH reached out to me because I was going to get a grant associated with the Illuminating the Druggable Genome Initiative. And she was involved with that, and they wanted to know if there was a conflict of interest. And I
wow.
was like, what are you talking about? And they're like, Lori Isom, she's consults, on this. I'm like,
Wow.
nope, no relation whatsoever. So since then we've met several times and we become good colleagues.
That is awesome. I love that story. That's why you told me let's hit record and then you can I can ask the question. This is really
Yeah, what are
cool.
the odds, right? It is an unusual, it is a rare last name. But to add, when we moved to Miami, my son at first he was in middle school, but after a few years he was in high school, and there were two sisters with the last name Isom,
Okay.
and everyone thought he was their little brother.
That's awesome. That is
So,
awesome. That is really cool. All right, let's talk a bit about science. And I'm very interested in hearing your story as a scientist. Did you always know that you wanted to be in science?
I was thinking about this today, and I can remember as a young kid being very interested in science and wanting to be and thinking about being a scientist and interested in scientific things. But I was also really good at drawing early on. And so that caught, the attention of those around me. And so this life of art really was began to just develop, throughout grade school and middle school. And that was my focus. I also played a lot of, I was athletic. I played a lot of sports. I really like sports. Um, and so, really, I was a late bloomer when it came to science because all through high school, I was focused on fine art. And when I graduated from high school, I actually went to art school. I had a scholarship to the Cleveland Institute of Art. I was born and raised in Cleveland. And at the time, I was also playing in a pretty popular band. And so it just worked out that I could play music, and do the art. And while I was at art school, it was in the University Circle area. And for any of your listeners that are familiar with Cleveland and the University Circle area, that is the cultural epicenter and the intellectual epicenter and the medical epicenter, where the Cleveland Clinic, Case Western Reserve is there, the Cleveland Orchestra is there, the Museum of Art. And so that's where I really developed, my formative years in intellect. And my majors in art school were sculpture and painting. And the way that CIA was run, it was 2 foundation years and then 3 years in your major. But to pay the bills, I had a minor in medical illustration. And for medical illustration, I had to do something they called cross register. And so I had to take science classes at Case, Cleveland State, John Carroll University, whatever needed to be done. And I was really good at them, and I had never had them before. And I was really interested in them. And so I secretly decided that I wanted to switch out of art into science, and I didn't tell anybody, and I applied to Case. And I got in, and my parents thought I was crazy, but I did it, and I switched, and I transferred to Case. I mean, they were scared too, because I'm first generation everything, first generation college student, first generation PhD, academic. They had no idea what I was doing. But I switched. I got a double major in chemistry and biochemistry. Um, my first year, I graduated in 3 years. My first year, I was lucky. I got a dorm room that where I could close my door and trap myself, and I didn't have to listen to anyone else. And that was my catch-up year, because I
Okay.
really had been focused in high school on art. And my dad was a janitor at my high school, and I could remember asking him to get me math books so that I could catch up on some of the mathematics I needed. And I just spent that year just, grinding.
Wow.
And and then the summer came, and you could take organic chemistry in the summertime, both semesters. And I learned that if I did that, then I could leapfrog, and that opened up all the other science classes. And so that's what I did, and that's how I got out in 3 years. It also made all my friends mad because they came back in the fall, and I was done with organic chemistry.
Wow.
Um, But that was a turning point for me, organic chemistry too. I thought I wanted to be a physician, but when I got into the organic chemistry, that really compelled me towards the basic research and what I eventually ended up doing.
What an interesting story. I do you still draw or sculpt in your free time, or do you ever use art now?
I There was a time when I could sneak some drawing and doodling in, and it's a great stress reliever. But once I started building my family, there's less and less time.
Yeah.
I told myself doing the science would give me some downtime to do some art. Yeah. So
Who are you kidding?
how naive was that, right? But what I've found is that, the computational stuff I do at the end of the day ends up being highly visual, a lot
Yeah.
of network-based connections, structure-based connections. And, the figures that we make often there's comments on, how they're assembled and put together. And so I think that that's my outlet,
Yeah.
right now for that sort of thing. And, I definitely get the bug every once in a while, though, just to like, oh, I wish I had a little place where I can go and like,
Yeah.
with an easel and set up shop, but not yet, not right now.
No, I feel you. I have zero art skills, but I do like to paint by numbers because then I don't have to think about it.
Yeah, yeah,
And I've done in 4 years, I've done two. The third one, I think I had to go buy the kit again because the paint dried out from the first one.
yeah.
And then I finished it, and then I re-gifted to a friend of mine the remaining of the paint, which obviously was enough with the Canva. But yes, I do understand the lack of time.
Of course, right? But there are two things in my life that are kind of like a mental wormhole where I can sit down and 8 hours will go by and I won't realize it. One is coding, and the second is painting.
Wow, that's amazing. That is amazing. And I actually forgot what I wanted to ask. No, actually, I wanted to make a comment about art in general, or just art and science. I feel like in my case, when I used to work in the lab and I used to look at the cells under the microscope, I mean, that is just so beautiful when the cells are
Mm,
healthy. And then I've I've looked at my hex cells so many times, or any other cells, that I could tell just by the way the light from the microscope was going through the plate if they were happy or not. And that's so satisfying. Or a beautiful dose response curve. And you're like, I couldn't even come up with the numbers to fit it this way. And the response of your cells gives you that.
No, you're absolutely right. I kind of was on a run of collecting old like TEM books of images of cells and Golgi and the mitochondria,
Yeah.
just because aesthetically their composition is so interesting and often
Yeah.
just so naturally balanced.
Yes.
And I mean, they have all these competitions now, right, for like best microscopy images,
Yeah.
and they're just stunning. And even the field of medical illustration, right, and what they've done there have just far surpassed anything I imagined,
Mm.
they would ever be, for developing and conveying understanding. These movies that you see like in some of these labs, like I know the Brian Roth lab, they put out several of these movies, and they're not the only ones. It's just what comes to mind
Yeah.
of these molecule GPCRs agonists
Yeah.
coming in. That just didn't exist, right?
Yeah.
And it's
Yeah.
it's a hugely useful tool for teaching people at all levels.
No, I agree. I agree. Which reminds me that there is, I can't remember her name. I think her name might be Piper. She's in Australia and she has all sorts of videos like this where she illustrates molecular interactions. It's really cute little movies where you're like, oh wow, I didn't think about my receptor this way, or I didn't think about this interaction this way. And that to me speaks to the artistic side of science.
Absolutely. And the name's familiar. And that probably is what I would have been doing had I stayed on the artistic path, I think. That's
Interesting.
that's what I would imagine.
I wanted to ask before we move on and talk about how did you get into studying GPCRs, because this is the Dr. GPCR podcast. I'm very curious to hear more about the medical illustrations that you were doing. What kind of work? What does that look like? What
Well,
did that look like?
at the time it was primarily hand-drawn or airbrush images that were meant to convey any concept that a medical student or a student would need to learn. So you've seen some of these, old, like maybe you've done a dissection of a cat and you have like, or a frog, right, in elementary school or middle school or secondary school, and you have these books with these drawings, right?
Yeah,
That is the most basic type of medical illustration as a guide. So you're an
interesting.
educator. Um,
Wow.
and you can, and it traverses scales. So you can have people, that's why you had to take the courses. You had to understand cells from molecules to organelles to lipids to
Yes.
tissues. And you had to use, it's where art meets imagination, because you're trying to convey, you know, you're trying to accurately convey the science, but in a way that you can digest the information. For instance, that's why we're not all pathologists. You look at, right, you see these talks, and you're not familiar with the cell type or the tissue, and someone says, look at all the lumen, and you're like, what?
Yeah. What does that eat in the winter?
What? And so you'd take information like that and you'd find a way to clarify it.
I love it. So art led you to science. What led you
Art,
to GPCRs?
ooh, what led me to GPCRs? There's still a long way to go between finishing the art and getting to GPCR. Should I fill in that gap?
Of course, please. Yes.
So I graduated from Case, and then I really wanted to fold proteins on a computer back before it was cool.
Now it's really cool.
That's really cool. And there were a few places where this was being done, but at the time, the best place was Johns Hopkins. And so I really wanted to go to Hopkins. I'm pretty sure I barely got in. I believe I had a faculty advocate. I mean, I know this now, I didn't know this then, right?
Yeah.
That really pulled for me. And it's just a great example of how one person can absolutely, change your life. And so when I went to Hopkins, I thought I would be folding proteins on a computer. For all those grad students out there, I didn't get my first choice of lab when I was doing my rotation. I got my second choice, and that was protein electrostatics with a guy named Bertrand Garcia Moreno. And so I really worked at a very reductionist level, studying how protons regulate protein structure and function. And that's what my PhD was in. And so what I wanted to do then is to parlay that skill set into protein design. And I wanted to design proteins that harvested energy
Mm,
before there were any real initiatives for alternative energy. So this is 2006. Right. And so there was really nowhere to go to do that other than a photosynthesis lab. There weren't like these institutes where, focused on alternative forms of energy or harnessing energy from biological sources, I should
yeah.
say. At that time, the Ventnor Institute had a small program on biohydrogen, but they weren't, they were keeping it small. And so I decided to do a postdoc in protein design, thinking that I could put those skills together and begin doing it myself. And so I went to Duke to do that under Homa Holenga. This guy was a protein designer. He was in the biochemistry department there. And he was leading a big DARPA consortium that included David Baker and others. Homa was the one that had started it. He had his claim to fame was that you take a protein of a known fold that's not an enzyme or
Mhm.
doesn't bind something, you treat it as an engineerable scaffold, and then you do some calculations to convert certain residues to actually bind or do the chemistry you want. So you're just treating the fold as a scaffold and then taking it over. And so they thought they had some success in the lab, and that was before my time. And when I joined that lab, uh, uh, I was tasked with trying to miniaturize the biophysical assays I had been doing, ligand binding, protein folding, things of this nature, to do very high throughput screening of protein designs.
Yeah.
And unfortunately, during that time, some of the previous work had been called into question and it had to be retracted, which caused all sorts of problems.
Yeah.
And so I managed to get two quick manuscripts related to these techniques out of that postdoc. But one day Hama came in and was like, "You and others just, you got to leave soon." And that was a big life lesson for me much later because it seemed like the worst possible thing that could have
Yeah.
happened for because I had this dream of opening a lab,
Mm,
right? And it seemed like the worst possible thing that could have happened. But in retrospect, it was the best possible thing that could have happened because I would never be sitting here talking to you with the career I have had I not had to take the next step, right? So
Comfort
this
out of the
happened
comfort zone, right?
out of the comfort zone. It was the Great Recession. I had a family. I had two kids. I had bought a house. It was limited options. And I meet Henrik Dolman at the University of Chapel Hill, and he's just like an angel there. He's a wonderful person, a wonderful mentor, and he is very open-minded. And from the get-go, he felt like I could bring very unique things to the field, right? That he didn't know what, I didn't know what, but if I just was familiar with it. So that is how I got into GPCRs. I moved a few miles down the street from Duke to UNC in Hendrick's lab. At that time, he was in the biochemistry and biophysics department,
Mm,
but now he's chair of pharmacology there.
yeah.
Top pharmacology program in the country, I believe, um, by the metrics. Um, and but it's weird. He works in yeast, right? He studied GPCR signaling in yeast. Yeast have one GPCR pathway for your listeners that aren't familiar. And many of the fundamentals of GPCR signaling had been discovered in yeast.
Yes.
Um, uh, Gap proteins, MAP kinase cascades,
Mhm.
and everyone was surprised that they were conserved in human cells. Um, And so I had been working on developing software to identify proteins that were likely to be proton sensors.
Mhm.
And so my first day in his lab, no joke, I downloaded some G alpha structures from the PDB, did my calculations, and I walked into his office and I said, I think that G alpha subunits might be proton sensors. And so spent the next several years proving that, and public and developing the yeast and biophysical systems to do that. And in the end, really kind of carved out a very unique kind of niche and way forward, and had expanded my knowledge base and skill set from just doing, protein chemistry and biophysics to doing, systems biology, to doing the yeast work, to getting into cell culture. And so I really had begun to be able to expand across scales.
Mhm.
And all this time continuing to code and develop structure-based informatics algorithms that I was using to make predictions that then we would test experimentally, either at the biophysical or cellular level.
I love
And so,
it.
yeah, and so he gave me the space to do that. And then,
How did you meet Henrik? Or when I know you mentioned that you were in this position where you had to leave your lab at the lab at Duke. Obviously, it's the worst thing that can happen to you when somebody says, "Well, actually, this is it," or
yeah,
sooner than you
sooner the
think.
better.
Exactly. Um, and obviously you mentioned having two children, having bought a house. I can only imagine the amount of stress that went with that. But how did you overcome that amount of stress and meet Henrik?
So, um, I've always, uh, I've always tried to stay optimistic, and I have the attitude that usually things turn out okay.
Okay.
So starting from that base, it takes help. It takes a village, right? Uh, Game recognizes game.
Mm.
My light went off behind me. Do you need me to turn it back on?
No, I think it's
Oh,
perfect.
good.
I think it's actually
Everything.
even
Well,
better because
it's a very green building, right? I
exactly.
have a very, I have a very cool and modern lab. So, um,
Love it.
Uh, so I mean, you need people in your life that help you make these connections. And for me, it was this guy at Duke, Terios, wonderful, wonderful biophysicist and MR spectroscopist, and he knew Henrik,
Okay.
and he knew and he had this feeling that we would work well together. And so he went the extra mile and put us in touch with one another.
Mhm.
And the rest was history.
I love it. That's one of the reasons I was asking this because the audience, and we were just talking about the Spotify wrap up of 2023 for us, but I do know, at least on Spotify, that the majority of our listeners are between 24 and 35. And this is the time where you're finishing up your PhD, you're doing a postdoc, you're moving on, you're building, we're starting to establish a career. And I always like to talk a little bit about, forming connections, being open to meeting new people, networking, or even finding a mentor, which is it's this big word, finding a mentor. I'm looking for a mentor type of thing. But this is the type of things that I think you, as a scientist or as a person, you need to be doing on a regular basis because, and you need to be helpful to others as well.
Absolutely. I mean, it's how the system works. I can give you a very clear example. When I was leaving Duke and that situation, I did try to do job, you know, I did try to enter the job market because I did have a few papers as a postdoc.
Mm,
And I just didn't have a community. I didn't have, I had been so stuck in just getting the work done, not making connections. I just didn't have that cloud of people that knew I was up and ready,
yeah.
right? And
Mm,
as soon as I got into Henrik's lab, and as soon as I got into the GPCR community, and as soon as I was going to meetings, right, it just I had a family, I had a
yeah.
group, right, and
Yeah.
an identity.
No, I think that's truly it. And I feel like that the GPCR community in general is very welcoming. It's diverse. And if you send somebody an email and say, do you have 15 minutes? I read your paper. I'd like to ask you some questions, or I'm planning my next step. And I was wondering if I could have you as a sounding board. I really like your work. I find you very impressive. Most people will say, of course, I have 15 minutes for you. Um,
Even further, I mean, I have colleagues in other fields that recognize that this field is far more open, right? Yes, everyone's concerned about being scooped about certain things,
yeah.
right? But in our field, I can tell you that, people are much more open than in many other fields. And it's very unique. And, I've even been in situations where people are like, no, I'm not going to work on that because this other person, I know they are, right?
Mhm.
There could be a natural competition there, right?
Yeah.
There's a
Yeah.
need to, but that's one of the things I really like about the field. It's healthy.
I think so too. I think so too. Especially, well, that was one of the motivations behind. I loved GPCRs from day one in undergrad in between 2000 and 2003 when Michel Bouvier was teaching signaling. And I was like, we had many other classes. But this, I am talking to you and I can see myself sitting in the bench in the classroom. And then actually at the time, I don't think it was this old school projector with the plastic, what's it
Yeah,
called
yeah.
in English? Um, and
Just the
Michelle was
transparencies.
exactly, and Michelle was drawing on that, and we were looking, and we had the molecular biology of the cell book, which was super heavy, and we carried it around all the time, type of thing. And from that moment, GPCRs were always a central part of my imagination. I think it's a as a system, it's a fascinating one, but also it comes with this group of scientists from all around the world. And when I started Dr. GPCR, it was out of a couple of frustrations. I was in a position and in a job that I didn't like particularly. I don't think he was fitting who I am as a person. And it was an outlet for me to create this podcast. One, to do something else, to keep my mind off of it, off of that situation. But at the same time, it was to get closer to the GPCR crowd, to bring the community closer. Because again, thinking to your position, I was in a situation where I didn't have a mentor. I didn't have a village. I knew a couple of people, but not enough for me to say, I'm in this position. I don't like it. I want to switch. Do you know of anything? Or can we have a conversation around what do you think I should be doing? Or you know me enough, in your opinion, what am I really good at? How should I be thinking? Because as in your case, when you were mentioning this, when you're in this stressful situation and you need to get yourself out, it's very hard unless you have a village, unless you have the positive attitude, or even the ability to say, okay, this is me in this problematic situation. I'm stepping out and I'm looking at myself and at everything surrounding me. And that's a very hard thing to do.
Absolutely. And you can't control the doors that will open for you. That's what your network does.
Exactly. Exactly. And I think being nice and kind and collaborative and in keeping in touch with
Absolutely.
people, and you know, the easiest thing is you read a paper from someone, you like it, just send them a note. Doesn't cost
Mhm.
anything. Makes their day.
Absolutely. I mean, you nailed it with the kindness. It's life's too short. We're
Yes.
all working very hard and
Yeah.
and that's how we, every day in the group and in the community. That's
Mhm.
definitely the hat we're trying to wear.
Absolutely. All right. So you're in Hendrik's lab. You finished or you're finishing up these projects, and how do you decide that you want to become a professor and not go
Oh,
into
geez,
industry?
I've already known that. That's why I'm still in the game. That's why I'm in my second post. I was a postdoc for 1, 2, 3, 4, 5, 6, 7, 8, 9 years.
Wow.
Yeah, I was
Wow.
hardcore. Yeah, and so I really wanted to have my shop
Mhm.
and have it be open to everyone and do some cool science. And so yeah, things were beginning, I it came to a point. I mean, I actually had a very difficult time getting a job. I had a very difficult time getting interviews. I really didn't have many interviews at all. And maybe some of that was due to my unusual path, et cetera, et cetera. But but it took one interaction, and I was at a Gordon conference, and this is the one that is out. Is it Long Beach or is it? It's the I'm blanking now. It's
Oh, Ventura?
the Ventura. Yeah,
Yes.
yeah.
Yes.
And Henrik was giving a talk, and it was related to the pH work we've been doing together. And Vlad Slepak, who is my colleague here at the University of Miami, came up to my poster and said, "Would you be interested in coming to the University of Miami?"
Mhm.
And I said, "Sure, absolutely, I'll interview." He's like, "Well, you have to have your application in a day or two because we're basically done. We've interviewed everyone." And so I said, "Okay, I'll do that," but I didn't do it. I was like, I didn't know how serious he was. And so he found me and he writes me an email. And he's like, "What are you doing? I need your application."
Wow.
So, um, so long story short, I went. I had never been to Miami before. Have you ever been to Miami?
No, never. It's
Yeah,
something I really
yeah,
want to visit though. I'm
yeah.
very curious.
Most people haven't. That was my first time, so you can imagine my shock. Um, and so I gave the talk, did my visit, got on the plane, and I had an offer for the position when I got off the plane.
Wow.
And you hear these stories of having to wait for months or whatever, like, and so very quickly they brought me back out, and the joke was I was 403. I was
Okay.
candidate number 403, and they didn't want to go to 404. I didn't know that until much later,
Hmm.
but it was that chance interaction that finally opened that
Wow.
door.
Wow.
Um,
403. So that means they interviewed 402 people that they rejected
well,
or
they looked at for, now being on,
applications. Okay. They
yeah,
didn't,
applications,
but
yeah.
still, I
Yeah, so
mean,
it's talk about feeling fortunate, right? I feel
yeah.
very blessed to be in this position.
I think it was about fit. They were looking for someone very
Oh yeah,
special
yeah,
or
yeah,
specific, and you were
yeah, yeah,
it.
yeah, yeah. And 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 it's been great. I mean, we can talk a little bit more about that, but uh, I guess the take home there is that I really knew what I wanted to do for a really long time. And so I was very motivated by that. And I was able to absorb some of the hits, but it was difficult. And I was definitely at the end of my rope. And going into
Mm,
industry wouldn't have been bad. That still would have been fun and exciting,
yeah.
right?
Yeah.
But I was right at that point. And if anyone listening is right there too, doors can open, like, you can
Yeah.
keep trying to make these connections and you just never know. I would have never imagined that I'd live in Miami.
Yeah. From Cleveland and then
From
to
Cleveland, yeah,
to
exactly.
North Carolina.
I never would have imagined it. And it's so exciting, and I hope everyone else can be as excited about where they're going next. But the key is I really knew, right? I wasn't on the fence.
Hmm.
And so that fueled me.
Yeah, I think that's really great advice to anyone listening. I think first you need to decide what is it that you want to do. What does that entail? Is that a lifestyle that works for you or not? And then put together a plan and then persist. You never know.
I'm not sure being a PI and getting a grant and getting grants is a lifestyle anybody wants.
It depends. It's something that I cannot see myself doing. I mean, it would drive me nuts having to write a grant. I mean, even writing papers was such a an extra effort. I would rather be in the lab designing, running experiments, analyzing data. I can
I hear
spend
you.
hours in front of Prism and looking at data and then playing around with it. But writing, no. I'm happy if somebody writes it and then I have to edit, not a problem. Love it. But if I have to start from scratch on a blank page, that is not an easy task for me. So
I have a I
please,
have a question for you.
yes,
So, Grammarly.
yes.
So I was like, I'll try this, just we'll see. I like trying, I do AI stuff, so I like to try out AI things for a while. So I'm like, I'll pay for Grammarly. I want to see if it's helpful. And so it's kind of just been there now and I'm used to it, but I got it in March. And one of
Okay.
the funny things is, it's not an advertisement for Grammarly, is that they send you these emails periodically and they tell you like, what you screwed up, like what you're good at. Like apparently the vocabulary I use is better than 95% of anyone else. So just I'm throwing that out there. Um,
Cool.
but then they also tell you how many words you've typed.
Okay.
So how many words do you think I have typed since March of 2023?
I don't know. I'm actually thinking between a very high number or kind of a very low number of, I don't know, 100. I'm just speculating.
What do you
All
think it
right.
is?
I don't know. Any clues? Can you give me any clues? What should I be?
It's higher than 500,000 and less than 10 million.
I don't know, a million?
It's 3.75 million words. I'm like, how? And I'm like, oh, it's my job.
Yeah, but
Emails, grants, papers, well, coding doesn't count. If coding counted,
then
it might be, I don't know, double.
wow.
Yeah. And I'm like, we almost do that. Not, I mean, it's
Yeah,
been since March, but that's a lot of words,
it is. It is. That's fascinating. So actually my
right?
husband had signed up to Grammarly. But I didn't know that you get these reports. I'm gonna have to ask him about his reports and see what does that look like. Wow, that's fascinating.
Yeah, so that's a small window into into the job.
Yeah, yeah. No, absolutely. And I think it all depends on whether what you want to do. And to your point, if you know what is it that you want very early on, then you have to stay the course.
Absolutely.
Let's talk a bit about GPCRs.
All
We
right.
were we talked a little bit about proton sensing and G proteins that you mentioned in the beginning, day one in Hendrik's lab. But what are you currently working on, and is it proton sensing GPCRs or proton sensing proteins,
Yeah, yeah,
or what
yeah.
are we talking about?
Good question. What are we working on now? So, I have I was actually part of the first cohort when I first came to Miami. I got an R35 Mira grant, and I was the first cohort of early stage investigators. And so the title of that grant was pH regulation of cell surface receptors.
Mhm.
So I've always been agnostic and it's meant to be agnostic, but really the focus has been G protein coupled receptors. We do have interest in RTKs and ion channels and other types of receptors and membrane proteins, but it has been GPCRs. Um, we're also very interested in how pH and protons regulate signaling pathways and components, and ligand receptor interactions in a very broad sense. Uh, and which leads me to, the problem. Like you had asked me earlier at some point what my favorite GPCR is. Well, I have a big problem because I don't know. I also can't tell you who my favorite band is, et cetera, because my brain doesn't work that way. So maybe this is just my global problem, but we're really agnostic and we work on too many
Okay.
GPCRs. Um, but if I was forced to pick my favorite, it would be the proton sensors.
Mhm.
And the reason for that is that, they were discovered 20 something years ago and they had histidines on the extracellular face. And so naturally, it's like, oh, those are the residues that are responsible for proton sensing. Completely reasonable, what's to be expected. And surprise, surprise, when you mutate some of them, the receptor doesn't work as well, et cetera. So the conclusion was that the proton sensing was, coming from the histidines. But this is a good example of how the lab works. We did a structure-based calculation, looking at the proton sensors and comparing them to all other G protein coupled receptors. And they had this very unique structural feature, which was this triad. It's a geometric construct. They're not actually in contact. Well, two are, but of acidic residues that were in the core, right? So they're not on the surface, they're deep in the core. Long story short, it turns out that those residues are what is responsible for the proton sensing of these receptors. And it's been such a fun ride figuring that out, and continuing to do those experiments. And, given what the mission has been, that's like a perfect win. We do a calculation, we predict the residues that are responsible for proton sensing. We make intelligent mutations to test them, and we're correct. That's the win for us in any, in any system. Um, and it's they're challenging. They're challenging because conceptually, like, when you search them in the literature, it's like they're involved in cancer, and one paper says they're good, one paper says they're bad. Right? They get endocytosed, they're going to turn on, right? Why? We don't know. And so they're considered dark receptors, pharmacologically dark receptors. And that is my favorite group. And that's where we've excelled. And that's where we've provided the most insight in the pharmacologically dark receptors, as defined by the Illuminating the Druggable Genome initiative. HCAR3 is another favorite one. Hydroxycarboxylic acid receptor 3. We discovered that a tryptophan metabolite is its endogenous agonist. And the fun thing about that receptor is it's only in humans and primates. Lower eukaryotes don't have it. So, and it's really important for fatty acid metabolism and immunity and other things. And that discovery has prompted a number of other studies and papers that have come out for the importance of HCAR3. But currently, we probably have between 70 and 100 receptors that we're actively studying. And we can do that because we're using both a yeast-based and then Heck or whatever based systems. And so our natural workflow is that we can do large pooled experiments in these yeasts that we've CRISPR engineered to contain human receptors and components. And that allows us to do, instead of being in cell culture, we're at the lab bench doing pooled experiments, just doing yeast work. And then when we have a finding, we then translate it out to Heck and then whatever cell type we need. We're using all the assays that everyone out there has created. A lot of Brett. You've done Brett.
And I'm Brett biased here. GPCR and Brett biased here.
Et cetera, et cetera. Yeah, so
you actually answered the question I wanted to ask, is since you mentioned that you work on so many GPCRs at the same time, what does that look like in the lab? Because obviously if you were to work on, people can spend 30 years working on the same GPCR in a hex system and then still discover things around that. But what does that, from a discovery perspective, what does that do? You always go full circle? So you try and predict something about the GPCR, about its function, its proton sensing capabilities on a dry lab format, then you test it out in yeast in a wet lab, and then when you discover something, then you can take that GPCR of interest, for example, and then go do a deep dive into that receptor in a hex cell system and do Brett and you name it? And then is there any information out of those wet lab experiments that go back into the dry lab aspects of it to kind of expand and build on that?
Yeah, that's a good question. Uh, we've been fortunate that it's been kind of a one-way road in that our predictions have been good so that we get to the destination that we
Mhm.
expect.
Yeah.
If we didn't get to the destination we'd expect, we'd go back. And that's a symptom of us still being a young lab. We kind of had a first big push of papers,
Mm.
and now we have the second big push that are coming out, and we have the time now to do exactly what you said, like look at some of this data, go back, refine calculations, go in and look at other aspects. Um, of their function. It's a balancing act, that's for sure. But the questions we're asking require a comparative analysis. Let me give you an example. So we made this discovery of proton gating of GPCR activation. And
Mm.
so I just told you about the proton sensors. They're activated by protons, and they probably have other endogenous ligands too that we're unaware of. But in this case, what we're talking about is we're talking about receptors that are activated by an agonist, cannot be activated by a proton, but the proton actually gates the ligand binding to the receptor.
Yeah.
And so what we found were that some receptors, some ligand receptor interactions are very sensitive to pH. And we found that some don't care.
Yeah.
We found that some are just graded. And so, in order to do that sort of study, you have to have the right system. So, for us, yeast is the proper system uh, to begin in,
Mm.
because when you change pH on the outside of yeast, we discovered that the pH on the inside does not change.
Mhm.
That's not true in a hex cell. When you change pH on the outside, you do get some change on the inside. So we wanted to just control for that. So what we did was we put 30 human GPCRs into the yeast system, and then we compare how their ligand-dependent activation changes as a function of pH. And we call this putting the pH into pharmacology.
Love it.
There are many receptors that we call Boolean,
Mhm.
like the dopamine 2 receptor, because it's on at pH 7, but as soon as you drop the pH a little bit, it's off. So it's almost like a step function.
Mm.
But then the HCAR3 is more like a graded receptor. As you drop pH, it gets turned off more gradually.
Mhm.
And then you have like the melatonin receptor that doesn't care what the pH is. And it just functions the same. You also have some receptors, mainly currently, that have carboxylic acid ligands that actually become more active when you lower pH. So we have this rich diversity of examples of how receptor ligand interactions and their synergy have developed this exquisite level of control that has just been missing from pharmacology. All drug screens have been done at pH 7.4.
Yes.
If you right, if you
Yeah.
take an inhibitor for the adenosine receptor, caffeine, and lower pH, it's no longer an inhibitor, right? And this has massive implications for when these receptors are endocytosed and then they experience the pH change within the endosome,
Yeah.
or an inflammatory microenvironment where you're acidified and you have the immune response, or in a tumor microenvironment. We're talking all about the same pH range band.
Yeah. Yeah. I find it very fascinating, and you're right, we don't yet have the answer to these questions. How is receptor function in ligand interaction and then signaling influenced by pH depending on where your receptor is expressed? And where does it would it signal differently once it gets internalized and it ends up in an endosome or a different cellular compartment where the pH is different?
Absolutely.
Love it.
And we're getting pretty good at this. I mean, some of the work that's going to be coming out in the next year, we should have a big dump again, is like the adenosine receptor. That was the only receptor that we discovered a proton itself can turn on. Again, we're in the yeast system, So if we don't add adenosine in, if we just lower pH, we get this nice sigmoidal curve. It comes on, it looks just like GPR68.
Yeah.
We can make very precise mutations to remove that pH sensing, but retain full agonism of adenosine. So you can
Nice.
imagine an animal where, collaboratively we use this type of, can is regulated by pH. Well, presumably we don't know if it's physiologically relevant,
Yeah.
and one that can't be, and say, is there a phenotype? Maybe there isn't. I don't know. But
I
those are
don't
the sort
know.
of experiments we
Yeah.
could do.
I love it. This is super interesting. Plus, I don't know much about any mutations specifically in the adenosine receptor, but maybe there are mutations that occur in humans that actually
Exactly.
shut on and off this response. That could be a really cool study to do.
Yeah, so that's something that we kind of look at too. We monitor Nomad and other big sets of exome and genome sequencing
Mhm.
initiatives to look for clues that way as well. Um, but at the end of the day, we're really kind of on the fringe. We're not working on the more traditional conventional aspects
Mm.
of the field, and I'm okay with that if the field's okay with that. you know and pushing um and I feel like a weirdo in that sense, but I think I'm not alone in this view. I think that in the next 100 years we're going to find that GPCRs do way more than the canonical G protein coupling and beta arrestin recruitment,
Yeah,
right? And this
I
is already
agree.
beginning to emerge. There's so much functionality hidden in the N and C termini of these things.
Yeah,
There are atypical chemokine receptors that only, suck up arrestin, et cetera, et
yeah.
cetera. And and I kind of am very comfortable in that kind of high risk area, like finding the new weird features of these
Yeah.
receptors.
I love it. I think it's important to keep an open mind and study these effects, especially developing tools to be able to measure these
Hmm.
becomes very important. And yes, you're right. Everybody does beta arrestin and G protein. Because one, it's relevant. I don't know how compared if we knew all the aspects of GPCR signaling and we had the ability to measure it and classify these pathways in a specific context. I don't know how much what would be the ranking of G protein signaling versus beta arrestin versus anything else.
Right, right.
But we've developed enough tools to measure G protein signaling and beta arrestin recruitment. Great. But you're right. Beyond that, what else? And how does the environment in general influence G protein, GPCR signaling in specific contexts? You can have a different cellular context, you can have a different cellular compartment context. And we need tools and we need to understand this. So I think you're doing really cool and interesting things.
Well, I appreciate that. It even extends. I really like to think about receptors that aren't even in humans, right? This is a huge family. And I maybe that's more what I mean. That maybe in what we know for humans, it's mostly G protein coupling, beta arrestin, and there definitely are other things that we can learn. But I just have this sense that there are far more exotic functionalities that we will slowly begin to peel back, that
Yeah,
will teach us, through an evolutionary lens, in other organisms, things that then again bounce back to humans, right? Just like all of the GPCR work done in yeast had
yeah,
relevance.
yeah.
Um,
No, I love it. I love it. Then I guess it's needless to ask if you think GPCRs are still good drug targets, considering what we just talked about.
I think that, not only do I think that they're excellent drug targets, I think that they could be used for so much more. For instance, are there certain GPCR expression patterns on tumors that can be used as markers, right? I
Yeah.
mean, they're very unique. There are pattern recognition opportunities in GPCRs. They're the most abundant family, right?
Yeah.
Um, you
No,
want to
no,
say something? I can
no,
see
I
it.
was just gonna say, yeah, and it's true. And one thing that we have sometimes don't have the tools, sometimes we don't have the antibodies to recognize these. We're talking about pathology and images. You need a tool to be able to stain your receptor. And the other thing I wanted to mention is splice variants are
Absolutely.
GPCR splice variants. Just they drive these guys drive me nuts because I'm always trying to figure out, okay, why would you have multiple proteins that are produced out of one gene that have different lengths, different, shapes, and then why would these signal differently, or would these interact together, or would there be a preference for a splice variant one to form a dimer with a heterodimer with another one? I think there's so many questions that I wish we could answer when it comes to GPCRs, but splice variants are the top of my list in there.
Yeah, mu-opioid receptor, over 20 plus splice variants in humans, 40 plus in mice, right? But most others just have like one or two or three, right?
Yeah,
That's the big winner, and it's like, what is going on there? And
yeah,
many of them can be detected in different cell types, as Pasternak's work, right? Um, uh, uh, but yeah, I mean, I also think that there are huge opportunities in biosensor design,
yeah,
in synthetic biology design. I think a huge opportunity, as pointed out by the Bill and Melinda Gates Foundation, is, fungal pathogens, right? Unlike bacteria and other microbes, fungal pathogens have GPCRs. This is how they mate, right?
yes.
And Chris Tate has the structure of sterile 2 from
Mm-hmm.
Saccharomyces, and
Yeah,
there are definitely real world impacts that can be had
yeah,
looking at these receptors in other organisms.
yes.
Um, I've already mentioned design, but I mean they're eminently designable for a variety of needs. I know, one group in Denmark, Michael Jensen, what they do, using yeast as a chassis is, um, they're trying to build synthetic pathways that make new compounds, and then actually have receptors that have been changed by directed evolution, some of the stuff we're doing too, that actually sense some of these, so that the system can sense when the product's been made, right? So there's this, just it's such a robust, you know uh prototypical fold capable of
Yeah.
so much functionality, that it comes back to the design I wanted to do in the first place.
Mhm. Really awesome. All right, top three aha moments that shaped your trajectory. Based on our conversation, I can pinpoint a couple that stood out for me, but I want you to maybe take a moment and verbalize those.
To me, it's more of a continuum. I I it's I feel like I'm not smart enough for aha moments, because every time I'm like, aha, this is a Nature paper. Then the next day we come in and we can't
Mhm.
repeat, right? So maybe I try to stay away from aha because I'm just superstitious.
Anything that you feel like might have changed the way you think about science or your work or anything?
Organic chemistry, really, really changed the way I viewed
Yeah.
science. It actually merged the chemistry and the visual for me, and then I discovered proteins in the same way from that. So that was a big transition. It was like, go to medical school or, go into basic research. Another aha moment for me was in my last semester of undergrad when I took C++ because I presumed that everyone in graduate school knew how to code. Then I got to graduate school and no one knew how to code.
I
And I
love
was
it.
ahead of the game, and that really helped,
Yeah.
that aspect take off. And then the other aha moment is like, I do my best work when I have balance in my life, when, yeah, I know my family's set like anyone else, right? And
Yeah.
it's and discovering that, finding that balance, and that's a constant battle, right? That's a constant fight when you're
Yes, it
passionate
is.
about anything, right?
Yeah. No, absolutely. That is definitely the constant battle because you want to be present. But then you mentioned, being able to sit in front of your computer and 8 hours pass by like this because you're so
Oh,
passionate about your work. But then there's people waiting for you at home.
oh, I get in trouble even at home. I can be very not present at home.
Yes, that's something I'm working on changing as well. Especially when there's something bugging me, I dream about things.
Yeah.
And then I wake up sometimes in the middle of the night and I'm like, no, no, it's midnight. You should be sleeping. And it did happen that I worked for 2 hours from midnight to 2 a.m. and then went back to sleep because I solved what I needed to solve.
I've been there. Yep. I
It's an
I
extreme, but it happens.
I have been working on code for literally a year, and I literally solved it at 5 a.m. The problem I did, well, I thought I'd solved it like many times, but
Yeah,
I
yeah,
knew that I did. And my wife went into labor at that time. It was a snowstorm in North Carolina. This is 5 a.m. I closed my laptop, which was a Mac, multiple screens open. I did not get back to that screen for 6 months.
wow.
And when I did, I confirmed that, life happened. I was
Yeah,
still working in the lab, I really couldn't take time, but that was kind of a side thing I was doing it was really the core of this Finder software I have, right? And that I was able to come back to it, and unbelievable
yeah,
how time flies. And
yeah. That's incredible. All right, before we let you go, two last questions. Any advice for junior scientists who want to contribute to the field?
I really, you have to know the fundamentals. You have to be able to connect to your peers, but at the same time, you have to have the courage to think outside the box and look at things from new angles, and to search for, what your niche could be or where the field can go. And, you know, imagination is a massive form of intelligence, and it can be practiced,
Yes,
and
and it should be. I think it should be
it should be.
practiced. And I feel like sometimes when you're younger, and I think pretty much speak of experience, when I was younger, I was so focused on learning what exists that there was little space to close the book and think about what else.
Yes,
And I think it was coming from a space where you're not born with confidence, you build that confidence. And so there is
absolutely.
this phase where you build your confidence, you build your knowledge, and that then you say, okay, I'm ready to explore and do something new.
Absolutely. It also happens when you start to give your first talks related to your research, right? You feel like you have to drag everyone through everything that you've been through instead
Yes.
of telling a nice,
Yes. Story.
and then you finally gain that perspective, and then you start to think like, oh, in bigger terms, and you can
Yes.
step away.
Yeah. Which it's something that I typically do. So we do have volunteers within our team, and in exchange for the work that they do, which I try to limit as much as possible because these people, these volunteers are PhD students or postdocs. I make myself available to review presentations, to review papers, to review whatever I can. And I'm brutally honest with them. And I keep telling them, why is this here? Is this important? What's the story? You just lost me. And I do understand. And I had the same problem as a PhD student. You want to tell everything that you've done, but then you can't drag everyone through every pipetting experiment that you've done. You have to tell the story because people will remember the story
Absolutely.
and not the details. I mean, details matter, don't get me wrong, but you need to be able to hold the person's hand and say, well, now I'm going to tell you this story and we're going to deep dive into this. This is important because it helps build the rest of the story. And this is something that I think, unfortunately, is not taught. You
No.
have to make the mistakes and then get this. Or if you're fortunate, then you have a PI or you have a mentor who's gonna, at the first time you do this, tell you, wait a minute, no. This is great. You have all the Lego pieces, but how about having a table filled with Lego pieces? How about we build something that people can admire?
Well, you froze for a little bit, but I'm sure whatever you said was just so
I
inspirational. I
I was
just, you know,
okay. Well, I'll repeat it. I was
no,
talking about, when the way I think about a PhD thesis defense presentation in its most raw form is like 30,000 pieces of Lego on a table,
yeah, exactly.
right? And then the polished version of it, at least that's what you're hoping for, is a sculpture of something. I don't know,
No,
it might be the
absolutely.
Millennium Falcon or something that people look at and like, oh, yeah, you built this.
Aside from actually having data for your thesis, like how the talk goes is a really excellent descriptor of your level of progress,
Yeah.
right?
Yeah.
As you mature.
Yeah. No, absolutely. Absolutely.
We all struggle
I
with it, though.
we all did. I was a total mess at my thesis defense. 20 minutes in, I had to apologize and say, I'm sorry, but I have to start from the beginning. And they allowed me, and I started from the beginning. And it was the worst thesis defense, at least in my opinion. But then I really did well at the question part where someone in the audience put up her hand and she said, "Can I play? This is fun. Can I ask questions?" And I said, "No, let's talk about it later. Let's just get this over with." But then, it's not, doesn't end up on your PhD diploma, messed up the presentation, started over again 20 minutes in. All right, then last question. If and when you have open positions in your lab, where can people find you?
Uh, I usually will put information on Twitter or the website, or we have the Blue Sky account, or I'll just kind of reach out to the network. I guess I
Mhm.
should use LinkedIn more. I guess I don't.
Yeah,
Uh,
you should use the Dr. GPCR
Dr.
Ecosystems
GPCR, yeah, I
job
get,
page.
yeah,
It's
job
just
page.
we do have a job page. You can just use a Google form in the ecosystem, and then we'll put up the information. We're actually in the process of revamping our job page and categorizing the job ads. Right now we have boxes, and the newest go at the top, but it's a mix and match of industry and academic jobs. So we're working on revamping those. And but 2 months ago, we started collaborating with someone named Mark Schmeisel, who's actually a recruiter for industry, and he's become our chief matchmaker at Dr. GPCR.
Very cool.
Really cool guy. Yeah,
This comes back around to having a community, right? People need to
yeah,
know who's up and ready.
exactly, exactly. Super, Dan. Thank you so much for your time. Don't go anywhere when we stop recording. We're gonna let people wonder what we talk about afterwards. But I had a lot of fun chatting with you.
Likewise, this was awesome. Thank you.
Thank you. Can I ask you a favor? Please subscribe to our YouTube channel. Many of you come back and watch our videos but aren't subscribed. Having more subscribers will help us get you more content. Also, mark your calendars and join us for the inaugural Endocrine Metabolic GPCR's meeting taking place April 22nd to 23rd this year in Birmingham in the UK. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guests, our Dr. GPCR team members, Attila, Ines, Montserrat, Ivana, Andreina, and Valint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials and subscribe to our monthly newsletter directly in the ecosystem. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Don't forget, we're also starting up the Dr. GPCR University. So if you'd like to join us for Dr. Karen Atkins' class, or you'd like to teach a class, please don't forget to reach out to us at hello@drgpcr.com. Until next time, stay safe.